a little bit on this personalized medicine idea, which is, can we predict in advance which particular mutations make a patient well-suited for a therapy or poorly suited for a therapy before they receive those therapies? And so with that, I just want to thank the people who've been brave enough to join me in the new lab, and also collaborators, both from before and current collaborators, and our funding sources, and probably the most important slide for this conference, which is this one. So if you're interested in a postdoc, we're recruiting postdocs too. So thank you.
Thank you very much for your talk. Now some questions.
Thank you for the awesome talk. I was wondering how many of the mutations that you get with these screens would be actually lethal on their own, like especially for the non-coding gene regions versus coding regions.
Sorry, say that one more time. How many of the mutations fall where?
They would be lethal on their own. So how many of them do just lose completely? Have you ever had estimates for that?
Sorry, how many? You're talking about the non-coding screen or the?
For both, actually. That would be interesting to compare. So if you have like RNAs, guide RNAs that would already kill the cell on their own, do you have estimate for that?
How many guide RNAs kill the cell? Is that what you're saying?
Yep.
Oh, yes. So a lot of people, us and a lot of other groups, I didn't show any of this data, have actually used this to try and figure out what's kind of the gold standard essential genes, meaning genes that, that if you knock them out of the cell's not able to survive. And when you do that kind of stuff, just the genes you'd expect are actually super enriched in that way. So for instance, any gene that's essential for transcription or translation is depleted within days. So you do get that. And that's been actually a difficult thing to map in human cells. What genes are, I mean, it seems like a really basic question, right? Like what genes are essential to life? But it's been difficult to come up with the gold standard set. Before, I didn't show any of this data, but previous ways of doing these genome-wide assays, like genome-wide RNA interference screens, because with RNAi, even though it's been able to discover quite a bit, there's a much larger degree of off-target effects than what we have to deal with these CRISPR nucleases. And so there, it's actually been quite difficult to establish a reference set of essential genes. Did that answer your question?
Yeah, pretty much. But then if you look at the non-coding screens for non-coding regions, does that also happen often?
Oh, do they also have a depletion end? Yeah, that's a really good question. So there are things. So there I only showed you the enrichment side. We actually didn't spend much time looking at, we spent a little bit of time looking at the depletion side. There's less stuff that's on the depletion side. So there you actually don't need the vemurafenib arm. You just look at just grow the cells for, say, 20 cell divisions and say which particular guide RNAs are less well represented. It's not so much. If you remember the control distribution, it was very tight. It doesn't change that much. Whereas in the genome-wide protein coding screens, you see tremendous dropout of certain genes because those are the ones that are essential or confer some kind of growth defect. Yeah.
Thank you.
Good question, though. Yeah. Sorry, it took me so long to understand it. You're loud. Yeah. Good.
Guides that are effectively killing the cell are also correlation with the open regions of the genome. That might be also that because of the guides are much more effective in these open regions.
Yeah, that's
controlled by effectiveness of.
I always get this question. Yeah. So I'm just going to restate your question so everybody hears it. So the question was, you showed that the regions that are enriched in vemurafenib resistance correlate with open chromatin regions, but that could just be because CRISPR, the Cas9 accessibility is higher in open chromatin regions. So there's a few things to say. So if you look kind of just at the papers, just take them at face value. There's one actually just published this week in PNAS from Dana Carroll's group that actually, I think the title is like Cas9 accessibility is affected by open chromatin, so it's very directly this. You will see a difference with open versus closed chromatin. The numbers range from like insignificant in the literature to like maybe a 10%, 20% indel difference. But almost all those studies actually are quite different than our study because what they do is they deliver, transiently deliver either protein, RNA, or plasmid. And we're doing none of that. So I didn't go into the details of the delivery, but we actually deliver this with a lentiviral method, which integrates in the genome, and then Cas9 is there basically forever for that cell's life. And so it doesn't just get one opportunity to cut or a few days to cut. Over the weeks that we do this screen, Cas9 is constitutively active in these cells. And so, compared to the indel rates reported in those papers, our indel rates are basically to saturation. So all alleles that are available to cut are cut. So it's still possible there is some effect there, but it's in the data in our deep sequencing data, when we sequence the target sites, we don't see that. We don't see a dependence on open versus closed chromatin.
In your non-coding screen, did you consider enhancer redundancy? And if so, what did you do to accommodate for that?
That's a good question. Did we consider enhancer redundancy? We didn't, actually. Very specifically, we didn't, because the way the screen is designed is that each cell just receives one of those small manipulations, right? And so you're saying, what about super enhancers where they're supposed to be, it's not like an additive effect, right? This whole idea of the super enhancer is that this entire large region is important. And so there's, I think, other ways to address that quite well. So one thing that some people have done is combinatorial screens, where you can deliver with either Cas9, or maybe a better option there would be CPF1, which is a naturally easy to chain multiple guides together CRISPR nuclease. CPF1, also referred to as Cas12a in the super confusing CRISPR literature. So you could deliver multiple guide RNAs to one cell. That's one option. The other way that I think might be better for addressing the super enhancer issue is this other technique, CRISPR-I, the CRISPR interference. And there, the nuclease function of Cas9 is kind of mutated out, and Cas9 is just used as a pointer into the genome, and tethered to that pointer is a CRAB domain, which basically has a, say, 500 base pair footprint of inhibition. And I think that might be a better way to kind of look at super enhancers, because you can kind of inhibit the whole thing at once. But also the whole idea of doing combinatorial screens, that's just starting in coding regions. And so I think it's very forward-thinking to suggest that for non-coding regions. But I think there might be one paper that's done that. But it's a great idea, yeah. There's all sorts of technological ways you could do that, just to keep going. I mean, you could make deletions. There's so many neat things you could do. That's why these genome editing tools have just opened a whole new world. Yeah.
So I have also one question.
Yeah.
How random is these CRISPR screens are? For example, comparing to radioactivity, because for a CRISPR screen, you still making the guiding RNA on your own, so you kind of connect it to this RNA when you do the screen.
So let me see if I understand your question. So you're saying how variable is the outcome in the genome? Is that your question? Like, what is?
Yeah. How random is the screen basically?
Okay. So this gives you some idea of how variable it is, and the question is, it is variable. Now there's other things you can do, like we mentioned CRISPR-I, or base editing would be another thing that could be tried. But as I mentioned, what these particular guide RNAs do, this is showing you data just from one guide RNA. It creates a distribution of indels in the genome. So we have thousands of these guides in our library, and each guide is actually seen by thousands of cells. So it's not just a single cell getting one guide RNA. And the genome editing outcomes are variable. Most often you see them kind of centered around a small deletion, like a 1 or 2 base pair deletion. But you also can see that there's a spectrum toward larger deletions. And this is, of course, what we can detect through PCR, through sequencing of small PCR amplicons. So there's probably, this tail is probably much longer. There's a few probably super long deletions we can't detect, and also some insertions. But you can see for a few different guide RNAs that there is a diversity of genome repair outcomes. So that's why I refer to this as mutagenesis. Did that answer your question?
Thank you.
Yeah. Great. Thank you.